Hello everybody, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we need to complete a thought, if you will. In the previous couple videos, we've been talking about energy metabolism, and our entire focus has been on breaking down glucose and then using that glucose as a source of electrons, as a source of protons, and to make energy. And so we're going to do a quick review, but then we need to get into organisms that don't use glucose or they're not using oxygen. And so we have these alternative mechanisms of energy metabolism, and that is our focus in today's video. Now, just a reminder, if you have not seen the previous videos on metabolism, please check those out. It's going to make this explanation a whole lot easier. I'll put the videos in the upper right-hand corner as well as in the description below. But we focused on glycolysis, transition, TCA, and then what we did with these steps is we made a table, and this table was basically all of the important things that we needed to get out of those processes. And so the substrate level phosphorylation ATP, that's the SLP. We tracked reducing power in the form of NADH and FADH2, and we said, okay, what is happening? What are we starting with? What are we ending with in each one of these steps? We also took a look in the last video at the electron transport chain, and we talked about how that functions. And one of the things that we talked about is when we break down glucose and we get all that reducing power, we send the reducing power in the form of NADH and FADH2 in the lower left-hand corner, and we establish a proton gradient that is then used to make ATP via oxidative phosphorylation. As we start to move forward, one of the things that we talked about here was that we have oxygen that is associated with complex 4. And when that oxygen serves as a terminal meaning end, terminal electron acceptor, the oxygen, along with some protons that are coming back in through ATP synthase, gets converted to water. This is aerobic respiration. And so when we say an organism is aerobic, what we're saying is that oxygen is being used as the terminal electron acceptor associated with complex 4 in the electron transport chain. So now that we have that quick background, again, I know I rushed through it, but again, I'm assuming that you've already seen the other videos related to metabolism. If you haven't, and I just confused you, please go check those other videos out. Okay, so we came up with a summary then, and we said if you have a glucose and you're using aerobic respiration, in other words, you have an aerobic organism that is using a chemical source of energy, glucose, and that chemical source of energy is organic, and therefore we have an aerobic chemoorganotroph. We've talked about these terms before. I will put that video in the upper right-hand corner and as well as in the description below. But if we have that, we can get a theoretical maximum yield of 38 ATP, but realistically, it's going to be less, often in the upper 20s to lower 30s. But the math worked out really well, and we can get a pretty good idea of what's going on in terms of ATP production with our aerobic chemoorganotroph. Okay, now that we have all that, let's take a look at some of these alternatives. Okay, so this is fermentation. And now what we're seeing here is we've got glucose at the top, just like I showed you in the table, being converted to pyruvate. We're getting 2 NADH and 2 substrate level ATP. On the right-hand side, if oxygen is present, then the reducing power is going to go to the electron transport chain. We're going to get ATP via oxidative phosphorylation. We've already talked about that. However, there's 2 other options. What if the electron transport chain cannot keep up with the energy demand? What if we don't have enough oxygen coming in? And so in that particular case, or there are microbes that don't even have an electron transport chain. Fermentation is another option for some organisms. And so what this is showing you is really a couple key points. During fermentation, glucose is being converted to pyruvate. That pyruvate is then being converted to an acid or an alcohol, and that's really a waste product that the cell can then release. Now, this is a really important point, and I want to go back to that table that we've been studying the last couple of videos. And so essentially, what we need to understand about fermentation is glycolysis only. Glucose to pyruvate. We just saw that in the previous slide. We get the 2 ATP substrate level, and we get the 2 NADH. But can we think this through for a second? If you have an organism that doesn't have enough oxygen or lacks an electron transport chain altogether, why would we ever need the NADH, and what is the point of further converting pyruvate to acetyl-CoA, and then eventually using it in the Krebs cycle. It doesn't make any sense to do that because the electron transport chain is not functioning or it doesn't exist. And so now the cell is left with a problem. I've made pyruvate that I can no longer utilize. I've utilized as much as I can. I've got reducing power that is basically useless because there's no electron transport chain for it to go to, or the electron transport chain is not functioning properly. But I did get the 2 substrate level ATP. So the issue, to be clear, is what do I do with the reducing power I don't need and the pyruvate I don't need? And there is a very ingenious solution to that. So let's go back to our picture. Look what's happening. The reducing power that was created in glycolysis is being used to convert the pyruvate to an acid or an alcohol. And so we've solved both of our problems. We've gotten rid of the reducing power, and we've also, in a sense, gotten rid of the pyruvate because the acid or the alcohol, and often sometimes CO2 is part of this as well, is now being secreted from the cell as a waste product. And so I always tell my students, when students partake of the alcohol side, really what they're drinking is microbial waste. And that makes people feel all funny and weird. So congratulations there. That's just another way to look at it. So people sometimes consume microbial waste. Congratulations. Okay, so that's fermentation. Now, the upside to fermentation, this process, is if the organism does not have oxygen available, that's great. It can still live. And so my motto for fermentation is it's better than being dead. All right, you're still getting the 2 substrate level ATP that we're seeing on the right-hand side during glycolysis. So you still get that, but not much more. And so what we wind up with is a net production of 2 ATP. So that is a far cry from the 38 that we are getting from our aerobic chemoorganotroph. So we've got a ceiling at 38 ATP, and now we've set the floor at 2 ATP via fermentation. Well, it turns out that life is very dynamic and very diverse, and there are other mechanisms that organisms can use both as an electron source and a terminal electron acceptor. So I just want to take a couple minutes and talk about those as well. All right, so what we have here is a really simple idea. In in a couple of videos, we're going to provide tons of examples. I promise, don't worry. But for right now, we're introducing some simple concepts that we're building on from a few videos ago. And so here we have some potential energy sources. On the left-hand side, we have a relative graph, and it is the ability of these substances to release electrons. Now, this is your energy source / your electron source. And so ideally what an organism wants, is to use electron or energy sources that release electrons very easily. After all, that is the whole point of using these things as an energy source. We want them for the electrons and the protons. And so what we see here, there are some substances, some compounds that release or give up electrons more easily than others. Turns out the organics, like carbs and lipids and proteins, they just give up those electrons really easily. That's great. We want that. And so for us as human beings, we are in a great situation. For microbes and other organisms that are chemoorganotrophs, we're starting with a substance, a compound, a structure that easily gives up electrons, and that is awesome. But if you look at this list, you're going to see some chemical sources of energy that are inorganic, non-carbon based. In other words, they are chemolithotrophic sources. Here, we have examples like hydrogen, elemental sulfur, iron, ammonia or ammonium. And we can see that these chemolithotrophic sources become increasingly difficult to extract electrons from. All right, so their ability to release electrons, not so great. And so therefore, if we are comparing chemoorganotrophs to chemolithotrophs, we could say, generally speaking, that chemoorganotrophs are going to be a better energy source or / better electron source than a chemolithotrophic source. But that's only one side of the equation. We have to be really careful here when we're making these assessments. And so we've got the energy source side, but now let's go ahead and take a look at the terminal electron acceptor side of things, because that's just as important. So here, same thing, the ability of this structure, this compound, to be able to release electrons. Now, our goal here is different. We don't want the terminal electron acceptor to release electrons very easily. We want them to gain electrons. We want them to be reduced. And so on this side of things, the further down we go, the better the terminal electron acceptor is. And so we've got here at the top organics. Not so great. Pyruvate being an example. We just talked about when you go from glucose to pyruvate, you get 2 ATP. And so while you can use organics as a terminal electron acceptor in some cases, for some organisms, it's not a really great option. But then as we see, some of these anaerobic, non-oxygen terminal electron acceptors like CO2 and sulfate, SO4, or nitrite, NO2, or nitrate for that matter, NO3, they're getting better, but they're not as good as oxygen. Oxygen does not give up electrons easily. In fact, it's way in the upper right-hand corner of the periodic table. It has a very strong affinity for electrons, and that is why it makes an awesome terminal electron acceptor. At the end of complex 4, it's super efficient. It's ripping those electrons away. And so ultimately, when we're going to determine how efficient an organism utilizes energy, we need two sides of the equation. What was the electron source? And so that was this information right here. And what is the terminal electron acceptor? This information. I'll give you a really good example. Often I ask my students, okay, will a chemoorganotroph always produce more energy than a chemolithotroph? They often get tripped up on this because they're like, well, yeah, chemoorganotrophs, they use carbs, lipids, proteins, that's way better. The key here is always. Think about it for a second. If a chemoorganotroph is using pyruvate as its terminal electron acceptor, so it started out really good, but it's only using pyruvate for its terminal electron acceptor. It's only producing 2 ATP, and therefore, a lot of times, it's going to be a far less efficient option than most of our anaerobes and or chemolithotrophs. And so here's a basic summary. And again, we're going to put a little bit more teeth into this in a future video, in the next video, well, in a couple of videos, where we give a lot of examples, a lot of different combinations. Right now, we're just trying to hit the basics. So as a nice summary, what I would say is an aerobic chemoorganotroph that's using glucose as an electron / energy / food source, all means kind of the same thing, and it's using oxygen, O2, as the terminal electron acceptor. When it converts that to CO2 and then the oxygen's converted to water, we're getting that maximum theoretical yield of 38 ATP. So that's a really, really awesome combination. So the next thing is the anaerobic and or the chemolithotroph. Now, you could have an anaerobic chemoorganotroph. You could have an aerobic chemolithotroph. So there's a lot of diversity there in terms of energy metabolism. But the main point here is that when you have a combination of those things, one or both, you're most likely going to get more than 2 ATP, but less than 38. So they're going to fall somewhere in the middle. It's going to be a wide range. And then lastly, we have fermentation, right? Those organisms that are fermenting, that are going from glucose to pyruvate, that's our floor. That is the 2 substrate level ATP. And so that's a really nice way to think about it, a nice summary. And of course, right now, we're not talking about all the details of all the different combinations. That is well beyond the scope of this video. Okay, so I just wanted to give you those basics because now we have a more general understanding. Okay, what does it mean to be aerobic? What does it mean to be anaerobic? What does it mean to be a chemolithotroph? What does it mean to be a chemoorganotroph? And once again, we've talked about these things in a previous video. And if you haven't seen the videos that came before this on the metabolism playlist, highly recommend check that out. In the next video, we need to talk about the last type of phosphorylation, and that is photophosphorylation. I will put that video in the upper right-hand corner. As always, thank you so much for being here, and we'll see you in the next one.